much, everybody, for being here. We have today a very interesting lecture, I'm sure, by Gabriel Almeida, who is Brazilian, but who works in Finland. And I mean, this seemed quite interesting to me, but I asked him to tell us a little bit of the story of how he moved and how he's, before telling us about his work. We are very happy to listen to what you have to say, and the floor is yours.
Okay, so thanks for the invitation. I'm very happy to be here. I'm talking to you today from Jyväskylä in Finland. So I've been here for actually 5 years this week. And I'm going to talk a little bit about myself in the next slide, but I just want to let you know that I'll be talking to you today about phage adherence to mucus and how it mediates preventive protection against pathogenic bacteria. And it's a pleasure to be here in this webinar because I really admire the CURE project. So I was very happy to be invited to talk to you. And a little bit about me. So I might be kind of a wild card in the phage research groups because my PhD was on human innate immunity while I was in Brazil. But I have always been interested in phages and also in phage therapy. And it started, I can recall that after I was watching the BBC documentary about Georgia and also the Data Institute. So it was the Virus That Cures documentary that I watched at some point in my career. And I never forgot about phage therapy, and I was really interested in trying to do something like that. It was very hard to start something in Brazil at the time because I was just a student and no one was doing that there at the time. And then me and my family decided to come to Finland in 2016, where I started to work with phages properly. And I'd say that's been good so far. So I have been to Georgia twice already. I met Nina there. I heard about the CURE project there. And I really liked Georgia. And so far, if you heard about me about phage research, probably it was because of this picture. So this is a picture that we took here a few years ago about E. coli cells being infected by T4 phage using a Hero microscopy, or because of this historical review that came out last year about the phage use in Brazil, how that was forgotten, that I managed to gather information using older collections of medical documents there. But what I want to talk to you today, as I mentioned, is the phage adherence to mucus history. And it's something that has been developed here at the University of Jyväskylä, which is situated in central Finland. So you can see here our department through different seasons of the year. It's close to a big lake, one of the 100,000s of lakes that we had in central Finland. And I've been working with Dr. Lotarina Sundberg. You can see her here in your department page. And her group is focused on Bacterium pulmonare bacterium and its phages. And we do everything here. So from phage isolation from the field to the study of host pathogen dynamics, and also some studies about phage therapy in aquaculture. So our focus mostly is this bacterium. We have others, of course, but we try to do everything from the isolation to the characterization and application of phages. And the idea behind this project came from this bacteriophage adherence to mucus model, so the BUN model. It was proposed by Jeremy Barr in 2013, so not that long ago, just a few years after it started here. And what this model proposes is a certain symbiotic relationship between viruses and metazoans. So the idea here is that phages, at least some of them, they are able to interact with mucin fibers on the mucosas. And by doing so, they are enriched in the mucosas. So this binding to mucins are not kind of covalent or anything. They are very weak bindings. So the phage is not actually bound to the mucin fiber, but it tends to gravitate around it. So it creates this form of subdiffusive motion, which is not the conventional Brownian motion that phages would have in any solution. So through this subdiffusion motion, what the phage makes, what happens to the phage, is that it tends to go inside the mucosa layer. So it's kind of attracted to the mucus source. And then it can create layers of phages on the mucosa. And probably it happens for several phages inside our bodies and animal bodies and so on. And these phages stay there waiting for the host to arrive. So it's a very nice system, because if you think about the logics about a phage and a bacterium meeting themselves outside, so let's say in the environment, the chances are very small because they need very near atomic distances to be together so that one can interact with the other. So you might think that evolution might have favored ways for the phage to find better its hosts. And this is one of the ways, because the phage can stay on the mucosa waiting, and then the bacteria comes to invade the mucosa and cause disease, and then it gets infected by the phage, which is already there. So as the model proposes, it says that phages can be a known host derived antibacterial defense in ourselves, and might be something that might be blocking infections all the time, and we're not even aware of that. And it's very important that this model has been shown to T4 phage, and it was narrowed down that T4 phage has this specific Ig-like domain in these head proteins that makes it interact with the mucin fibers. But of course, it was on in vitro conditions, like artificial conditions. But anyway, we liked the model a lot, and then we decided to test that in a more realistic situation. And what we chose to use was Phylobacterium columnari. So it's a bacterium which causes columnari disease. And it's an opportunistic pathogen. So it can be found everywhere, so water, mud, or even the microbiome of fishes. And it can cause disease on fishes on certain conditions. So the triggers are not that well known, but of course mucus is one of the triggers, and also temperature is another one. So that's why here in Finland we have outbreaks of columnari disease every summer, for example, when the water temperatures goes above 20 degrees. And columnari is a very versatile organism, so it's very tricky and also exciting to work with. So first of all, it can survive in the environment for long periods. Like it can stay alive in pure water for several weeks, for example. It has at least 3 different colony morphotypes, depending on what's happening to the cultures and to the conditions. So on this picture here, for example, you can see this type of colony type and this colony type, and they are from the same strain. So they share the same genome, but you can see how differently it can grow depending on the conditions that you give to them. And it can also respond to very small variations in growth conditions. So it can sense small variations of ions in water, for example, and then it can change completely how the bacterium grows and whether it causes disease or not. So it's a very tricky organism to work with. And here, of course, we have the colonies, and here you have details of one of these rhizoid colonies. And here you have the cell organization in one of these tentacle-like structures that grows on the medium. And the disease caused by this bacterium is called columnari disease, and it typically affects the fish skin, the gills, and the fins. So it's a mucosal disease. So the bacteria doesn't go inside the fish; it just causes disease on the skin and on these other organs. And to reach the skin to cause disease, then the bacteria must cross the mucosal layer. So there's a very close interaction between this bacterium and the mucus of the fish. And we know that the mucus has a dual role. So at first it can be an attractant to the bacteria and also could be used as a nutrient source, but also it's part of the fish defense. So that's something physical, immunological here that the bacteria must do to start causing disease. And Lotharina group here has already shown that phages can be used for phage therapy against columnari disease. So here's some pictures from her group. And it's interesting because only the group here has these phages isolated so far. We don't have any other collaborator with these phages in hand. So we only know about these phages from Finland. And one of the reasons might be these mucosal interactions that I'm going to be discussing with you. But anyway, taking this all together, we thought that columnari disease is a relevant model that we could be used for studying mucosal diseases and also for testing this burn model in a real infection model. And then we decided to think about how to show that. And the first question and the first result I want to show you here is whether our phages bind to mucins or not, or to mucins and mucus or not. And the idea here is that we needed to show that we had the same system as Jeremy Barr when he showed this model. And the answer here is that yes, we had several phages which binds to mucins. So what I'm showing you here is a result from some experiments in which I took agar plates, either normal agar plates or agar plates with mucin on the composition of the media. And then I wash it on top of this plate some phage solutions. I let the plates dry afterwards, and then I counted how many phages were stuck on the plates. And the results is shown here on the y-axis as the counting of the phages, and the black bars are the control plates, so the conventional media, and the gray bars are the mucin-containing plates. And you can see that several phages have the tendency to be found more in the mucin-containing plates, which might indicate that they are interacting with the mucin fibers and stay retained there. So we tested, for example, one Bacterium columnari phage, one phage which infects environmental Bacterium species, and PRD1, which infects Salmonella, T4, which infects E. coli. It has been the basis for the model. Flip is a Bacterium phage, which is unrelated to the other ones. It's a single-stranded DNA phage, but it doesn't bind to the mucin. And V46 infects Aeromonas strains from the environment. So we could show here that several phages actually are binding to the mucin plates, not only T4 as predicted, but many others. And we chose phage FCL2 as our model because this is the phage that we had it best characterized. And then the next question was whether this mucin binding also happen in real mucosas. So of course, it's very different if you consider agar plate containing mucin, everything sterile and static there. If you consider an animal, for example, the gut of a mice or skin of fish and so on. Because if you go to the animal, then you have the effect of the microbiome and also mucus shedding and any other variable. So we did an experiment to show whether the phages are also being held in the natural mucosas. And for that, what we did was to take some fish, and we put this fish in an aquarium. In this aquarium, it had flow-throughs, so it means that the water was being exchanged like 800 milliliters per hour. So 800 milliliters per minute. So it was a lot of water changes. And once the aquarium was set, we then gave some phages to these aquariums, and then we left it staying there for a week. And every day during this week, we would go there and take the 3 random samples from the water and also 3 different fish. And the fish was being euthanized and then scrapped in order for us to get in the mucus from the skin. And then we were measuring the phages from the mucus and from the water during this 7-week period. And what I'm showing you here is the titer of the phages on the samples over time. And you can see that the trend is after you give the phages, you can detect a lot of phages in the water as expected, around like times 0. But then one day afterwards, the titers drop drastically. But after that, what you can see is that the phages tend to stay on the mucus, so that is the black lines here. And it's only found occasionally in the water after the first day. So that indicates that the phages are being enriched in the fish mucus and not just floating around in the water as they should if they are not interacting with the mucins. So we could show that yes, phages can bind to the fish skin mucosa in vivo. And the next question then was whether phage binding to mucus have any relevance for protection, like predicted in the model. And for that, we took fishes. So I didn't mention, but these fishes are rainbow trouts. So that's the model we have here. But we took them, and then we gave phages to them again. And we left them for a while with the phage, so 1, 3, or 7 days. So just 1 dose of phage in the day 0. And then we infected them with Bacterium columari at an lethal dose. And then we saw if this phage pretreatment could protect them. And just for comparison, we did 2 groups here, one with our phage alone, which infects columnario, and one also had phage T4 together, because we wanted to see if there was some form of competition for mucus space between the different phages. And T4 infects E. coli, so it's something that's not relevant in this model here. So it wouldn't affect anything related to FCL2, but it would have another phage competing with our own for the mucus space. And before showing the survival data, I just did the same as I did in the previous slide, which was to measure the amount of phages in the mucus and in the water from 2 different groups. And this trend does the same. So I can see more phages in the mucus than in the water as the times go by. And that happens for FCL2 phage in both groups and also for T4 phage. So we're also showing that T4 is also able to bind to primary mucus and not only to mucins in vitro. And then we evaluated fish survival after giving the columnari dose to them. And here is for the first group. So you can see on the dashed line our negative control. So these are the fishes which didn't receive any phage and any bacteria, so everything survived at the end. The black lines are the positive controls, so these phages only got the bacterium but not the phage, and then they died. Everything had died 34 hours after the infection. But if you see the line, if you compare the groups which had the phages before, so either 1 or 3 days before receiving the bacteria, you can see that there was a delay in the start of the disease and also a protection of some animals. So showing that this preventive treatment, so this previous dose of phages was enough to protect the fish from the bacteria which came afterwards. And this result, the result was even better if you check the mixed phage group, because then even phages which had, even fish which had the phages 7 days before infection were also protected. So that was something that we thought very remarkable, but sadly we couldn't explain why that was happening, because T4 is not replicating on the system, but having T4 together was somehow making our phage more efficient. So to us, first of all, it points out that for this preventive approach to phage therapy, phage cocktails might be good because one might help the other. And one explanation might be that T4 is larger than FCL2. So that means that FCL2 can go deeper in the mucus layer than T4, and T4 might be helping to hold the smaller one below, but it's just a speculation so far. And then we associated this survival data to the amount of phages and also the amount of bacteria in the aquariums, so that we could really show that fishes are surviving because we had more phages and less bacteria. So on the top graphs here, what I have is the quantification of phages on both groups. And you can see that, so time 0 was the time that we gave the bacterium. So you can see that as infection times go by, the number of phages increases, and that's dependent on the initial amount of phages. So the longer you kept the fish with the phages, then you had less phages in the fish. Then it took longer for the phages to reach high titers, but we could recover high titers of phages on all time points, on all pre-treatment groups, sorry. And if you check T4 levels, you can see that T4 is stable over time, so it's not replicating, so it's not affecting directly columnari probably. And then you can correlate these phage amounts to the number of bacteria. So here it was measured by the number of genomic copies by qPCR, and you can see that the bacteria starts to increase. But then, if you when phages hit high titers, the number of bacteria decreases, and that nicely correlates to the groups which were protected in the survival experiment. And our first conclusion was that the retention of phages by mucus was mediating protection. So if you give phages before to the animals, you can protect them for a bacteria that comes afterwards, because the phages stays in the mucus. And another question that we had was whether the mucin or the mucus also affects the bacteria alone. So far we're just checking mucus and phages, but then we wanted to check mucus and mucin and also the bacterium. And what we saw that it's very easy to see is that if you give mucin or mucus to Faelobacterium columnari, you induce very quickly the formation of biofilm. So normally this bacteria grows as normal E. coli, for example, just gets turbid over time in media. But if you have mucin in the media, then you can see that it quickly forms this biofilm ring. So it happens in different, you can scale this up, and it happens different temperatures and also in different rotations and so on. And it's dose dependent. So the more mucin you give, the more biofilm you end up forming. And that happens for all Faelobacterium columnari strains that we tested. So everything that we had in hand, we tested, and it happens to all of them. So that's something that's really widespread among these species. And then we wonder if this biofilm formation, of course, was leading to increased bacterial virulence. And then it was tested in different ways. So I guess I don't need to go into too much details here. But basically, if you give mucin to the bacteria, then you increase chemotaxis. You can also increase the size of the colonies on agar plates. And this might be related to motility, which is linked to the type 9 secretion system, which is a virulent factor. We can detect some differentiated colony types in the mucin cultures that never appear in other types of cultures. And we can also see that the mucin-exposed bacteria also produces more proteases. And finally, we saw that mucin exposure makes the bacteria more virulent in vivo. So the idea here on this experiment, so there were no phages on this experiment, only fishes and the bacterium. And what happened is that we infected the fish with bacteria which has grown on normal media or in normal media containing mucins. And what we saw is that if you need a dose which is like 2 times, if you give a dose, a certain dose of bacteria exposed to mucin is capable of killing better the fish than a dose 2 times higher than that, but grown on control conditions. And Azico was a dose which is 10 times as that, but grown on the control conditions. So in a way, mucin prepares the bacteria to be virulent, and then it can kill the fishes faster or more efficiently. Anyway. And the conclusion here then is that exposure to mucin or to mucus, it upregulates virulence factors, and then it increases the virulence in vivo. And finally, for this first part, what we thought is how does everything works together. So if you have the viral adhesion on one side and you have the upregulation of bacterial virulence on the other, but what happens if you put everything together on the same culture? And here I just need to let you know a few background, which is that growing these columnar phages is very tricky because the phages don't like to grow in liquid cultures at all. And the bacteria sometimes doesn't like to grow in the soft agar. So it creates a lot of problems if you want to have them grow and have high titer of the phages. But we tried anyway to mix everything together. And what we saw was very nice. And it's shown here for primary mucus and also for purified mucin. What happens is that if you check the control conditions, so this dashed line here, the phage is not growing on liquid cultures as is expected. But when you give a certain amount of mucus, for example, you can see that the phage grows very nicely. And that happens on the same trend for purified mucin as well. So we could see that somehow the phage prefers to infect the bacteria which has been exposed to mucin or mucus beforehand. So our idea here is that the phage can exploit this increased virulence to infect better the bacteria. And that was nice for the biological point of view, but also for allowing us to produce these phages in high quantities using liquid cultures, which wasn't possible until then. And after all that, then I tested a lot of the pulmonary phages we had. So we had a big collection here, and we tested almost all of them, and almost all tested ones are working like that. So if you gave mucin to the host, then the phage infects better and produces more progeny in liquid. And then the next step was to think about other phage bacterium pairs. So everything so far has been focused on colonari, but we wanted to see if that's something that was happening also in other groups. And then at first we tested what we had in hands. So we tested E. coli, and nothing happens to the phage numbers. Actually, the mucin present doesn't affect how T4 phage grows. The same for a salmonella phage and some Faelobacterium environmental strains, and also for some Faelobacterium pseudophilum species, because pseudophilum is another Faelobacterium which causes disease in fish, but it causes internal diseases. And in this case, its phages are not affected by mucins at all. So our conclusion at the moment was that mucus mediated upregulation of virulence and also the susceptibility to the phage is not widespread, but it's also not limited to columnari. And that's because at the time in our collection, we found this other bacterium, which are also responding exactly the same way as columnari had responded. So this is an Aeromonas bacterium, which comes from the environment. I have no data about virulence or anything, but it was very clear to see that if you give mucin to this Aeromonas, it forms biofilm, And if you make the liquid cultures, then the phages grows better if you have mucus or mucin present on cultures. So it's exactly the same as columnari, but it's an unrelated bacterium. So we start to think that you might find among different groups of bacteria different examples that behaves like that. And that might be some form of widespread way that bacteria and phages have to interact inside the mucosal layers of animals. And just to give some support to that, I just want to remember you about the first result I showed, that most of the phages we tested are able to bind to mucins. So it might be that they have different hosts that they can infect. They can also respond to the bacteria changed by the mucins. And then what I showed you here is already published on MBio, so if you want to read that in details. But we haven't stopped there. So, for example, very recently, I got some additional data concerning Pseudomonas aeruginosa, and that comes through a collaboration with Dr. Nina from the Eliava. And for me, that's very important because it opens up a lot of possibilities for phaging. So phaging is the oldest, or the old way of using phages as prevention or prophylaxis, and not just a treatment. And maybe this interaction with mucus and retention in mucus might be the link between the older phaging trials and modern phaging uses for phage therapy. So that's something that I really want to go forward in my research, to try to see if that's true and try to test it and show that it could work. Because the gain here, considering mortality, morbidity related to bacterial diseases, is very big. And on a way, we think that the mucosal surfaces or mucins, they can function as a transcriptional regulator. So the bacteria not only use mucins as energy source, but they're also using mucins as a way to activate virulence. And on this transient virulent state, then some phages can exploit and infect better the bacterium. And that ties well to the Bunn model, because the phages which are able to stay in the mucus are the phages which are able to better infect the bacteria which has been changed by the mucus. So it makes a lot of sense that evolution might have favored both, like this exploitation of the virulence and also the retention in the mucus. And then there's also some trade-off that we are not that aware of for the bacterium, because at least for some of them, they might be protected from the phage outside the mucus, but susceptible to the phage inside the mucosal layer. And of course, the trade-off is that if they are on the mucosal layer, they're causing disease, so they can have more progeny, they can have more energy, but at the same time, they are at risk of being infected. So that's something that we still don't consider this trade-off, but that it might be very important for disease outcome, and also for studying phage, bacterial, and hematozoa ecology and interactions and so on. And I have here this simple model that we made for columnari that might be applied to others. So columnari usually lives on the environment where they are safe and protected from the mucosal phages at least. But then if it senses mucus, it starts to change, upregulates virulence, and then starts to cause disease in the animals. And here it has a lot of gains regarding energy and progeny production, but also it has this problem that can be infected by the phages. And this susceptibility to the phages is related to the concentration of the mucus on the medium. But if the bacteria survives causing disease, then it goes back to the environment and goes back to the phage-protected, phage-resistant state, which are not causing disease in the fish. And it's, I guess, important to highlight that the here at the University of Vascular, we have developed methods for phage isolation, phage quantification, and also phage growth based on these mucosal interactions that have been discussed. So we have all these things well set, like ways of protocols of how to use mucins to make liquid cultures of phages, or how to titrate phages, and also how to isolate phages from the environment. And it's something that the university has patented, so it's important to make this disclaimer. And where to go from here? So getting near the end of the presentation, in time. First of all, I want to expand this model to other phage bacteria models. So that's something that I have been doing for a while, as I mentioned. And here are my contacts. So I don't have So I'm just putting my personal email address here because the one I'm using at the moment is going to be ended soon when I move to Norway. I don't have the one from there yet, so we can keep this one for now. And don't hesitate to contact me if you have any idea of collaboration or some questions or something like that. But at the moment, I can answer your questions, I guess. But thanks for that. Thanks for the attention.
Thank you very much. This was really great. Very, very interesting.